conferences, and we're so honored that each time he agrees to come, and this time he agreed to come to Columbus. So please give a warm welcome to Dr. Feagan. Thank you.
Well, thank you, Anita. It's not very far from London, Ontario to Columbus, so it's not much of a journey. I've been asked to give a forward-looking talk here, and you'll be delighted that I'm not going to go into the minutiae of every drug that's in phase 2 development. I'm going to concentrate on drugs that are going to come to the clinic with a high probability within the next 3 to 4 years. So I still have the beta version of the Microsoft crystal ball, but I think the vision here is pretty accurate. I'm not going to waste your time about things you'll never see or use. So these are my disclosures. Start off with a couple new monoclonals. And so anti-endocrine therapy has really gone through an amazing transformation. If we look back to the dark days of natalizumab and the crisis with progressive multifocal leukoencephalopathy that almost killed the vedolizumab development program. That drug came within a whisker of never coming to market. But we've come to understand that selective ability to affect T-cell trafficking is a really good strategy in IBD. And it's amazing, actually, that vedolizumab actually works because it only affects 3% of your circulating T-cells that are the gut. Lymphocytes still traffic into the gut through alpha-4 beta-1 VCAM interactions, even when MADCAM is blocked. So this is a new monoclonal, and it, instead of targeting alpha-4 beta-7, remember that vedolizumab doesn't block alpha, doesn't block beta, it blocks the shared epitope where the two come together. Well, this antibody is beta-7, so it would block alpha-4 beta-7, but it also blocks alpha-E beta-7 E-cadherin interactions. Well, what's that? You know how proud your pathologist is when they show you those intraepithelial lymphocytes in celiac disease? Well, the way that those T cells actually express alpha-E beta-7, and it's the protein that tethers them to the basement membrane. So that's why they're stuck there through that interaction. So this antibody will block alpha-4 beta-7, like Vedo, but it'll also block alpha-E beta-7. Now, is that good or bad? So this is less selective than vedolizumab. You could argue, well, it's going to be better because those cells are probably causing trouble too, so block them. So it may be more efficacious, but you could also argue, well, those cells are present on places other than the gut, such as the lung and the skin, and that may lead to increased toxicity. So there's an enormous program with this drug, probably the most ambitious clinical drug development program ever. A total of 7 trials, probably $2 billion worth of clinical research budget. And it's testing this hypothesis, and this is the cartoon just showing where this is working in the lymph nodes, in the tissue, and also in the epithelium, as distinct to Vedo, which is just the lymph nodes in the tissue. Okay. So we already have a phase 2b study, and this result was published 2 years ago in Gut. And you see this very nice result in an induction study where 2 different doses of the regimen were compared to placebo for clinical remission. And you see in the overall population that there was a demonstrable clinical benefit. I don't show you the data here, but what was interesting, this is probably your first positive biomarker study where the investigators showed that if you took patients and you measured beta-7 expression in tissue, the patients who had high concentrations of beta-7 in the tissue actually did better on this drug, indicating the possibility of using it as a diagnostic biomarker for patients responding. So this is in late-stage development. I'd say about 3/4 of the phase 3 program is complete. And there's also a direct comparison in this to a TNF blocker. So we heard this morning, Scott said there's no comparative effectiveness studies. Tune in about 2 years and you'll have a comparative effectiveness study with this agent. Now I'd like to really move on to something that's really exciting, I think, that's going to be transformational, and that's the notion of targeting 23. And you heard about ustekinumab this morning. You're familiar with it in the clinic. It's an excellent drug for treatment of Crohn's disease. It targets both 12 and 23. And this was a rather amusing story, I think, dating back to about 7 years ago when this development program was started. We got the idea that, well, it'd be a good idea to block interleukin 12, because interleukin 12 is the master cytokine that regulates the Th1 pathway. So, eliminate the middleman, don't block TNF, block upstream. He said this is going to be a good strategy, and there may be more toxicity if you block the whole pathway as just blocking TNF, but it's worth looking into. And we got into this at the same time as psoriasis, and the dermatology doctors managed to show an effect in 17 patients, open label study, and within 6 months they knew they had a drug, and 2 years later they were to market, and showing that it was highly effective and was more effective than TNF blockers. Along the way, we discovered that there was this other pathway called Th17, and lo and behold, the P40 subunit that we were targeting was shared by both of these. Th17 pathway, interleukin 23 is the driver, akin to interleukin 12. So I think our initial, when the basic scientists told us this, we said, well, it would have been nice to known that that's what we were doing when we set out on this path. And this will work for sure because you're blocking the two major inflammatory pathways, but gosh, there's going to be hell to be paid for toxicity. And in fact, the dermatologist sorted that one out pretty clearly, that don't ask me to explain it, but blocking those two pathways is perfectly safe. And we got the results from just recently, this study published in 2016, showing induction therapy with 2 different dosing regimens against placebo, and a very strong maintenance effect, as Scott showed you. Well, this set out on the table, well, if blocking 12 and 23 is good, what if you just block 23 itself? And I'll just as an aside tell you that we actually tried to exploit this pathway some years ago by blocking not interleukin 23, the driver of the pathway, but its terminal cytokine, interleukin 17. And that wasn't a good idea, actually. It turns out that unlike psoriasis, you've probably seen patients now on the new generation of anti-IL-17s. Some of those patients will develop Crohn's disease. And it's a debate whether that's just overlap syndrome or where, in fact, this drug is actually bringing out latent Crohn's disease in these patients. So the story in interleukin 23, as I mentioned, it's the driver of the Th17 pathway. And we did this study with risankizumab, which is I think almost approved now for dermatology. And so it's a selective monoclonal inhibitor of the interleukin 23 P19. And you can see this is a conventional dose ranging study, 600 milligrams, 200 milligrams in placebo in a 12-week induction study. And with this result, and this was in a group of patients, 96% of the patients in the trial had failed a TNF antagonist. So a very tough crowd. And what you see are the clinical endpoints of the 2 doses, and on the right-hand panels, you see the endoscopic endpoints. And note deep remission, that is complete mucosal healing and CDI score below 150. You don't see that in a refractory patient population. So this is a very strong signal, sufficient enough to have AbbVie purchase this drug from Boehringer Ingelheim, and it's now in phase 3 development. And there was actually a durable effect in the trial as well, showing good anti-inflammatory effect in maintenance. A result with another agent, this was a study run by MedImmune, again, a specific anti-IL-23, again, showing a really good effect in refractory patients with Crohn's disease in this gastro paper. So just to give you a sense of why this is exciting, these drugs are perfectly safe. They're as safe as ustekinumab. And in this situation in dermatology, the interleukin 23 antagonists and the anti-IL-17s are more effective than Stelara, okay? And statistically, head-to-head studies. So if you extrapolate this into Crohn's disease, ustekinumab, they only have a half-life of 3 or 4 years. So, this is a next generation, and I think this is highly promising. Tune in at DDW. There's going to be the first trial of an interleukin 23 inhibitor in ulcerative colitis as a late-breaking abstract. So this class of drugs, I think, is a game changer. Okay, and I told you about the IL-17 story. This is probably one of the most tragic studies I was involved with. I don't, when you have a negative study, it's one thing to have an inconclusive result, no effect. But this study was a trial of secukinumab which is Novartis's anti-IL-17. It's on the market. Your dermatology colleagues are using it extensively. And this is a study we did some years ago, looking back to 2012, where we piloted this in active Crohn's disease. And what you see here are CDI scores over time. And man, the key to the figure isn't switched. So the patients on drug statistically significantly had worse disease activity than placebo. And in a trial of 62 patients, we saw a statistically significant increased risk of serious infection in Crohn's disease. In dermatology, there's no signal at all for infection. So the gut is different than the skin. And we don't know why IL-17 is protective for the gut, but it appears to be. So direct inhibition of IL-17 is not a good idea, where the pathway driver, anti-IL-23, is highly promising. So we don't understand why that is, but those are the observations. I'm now going to flip over and talk about small molecules. And small molecules, I think, are the future. And those of you who follow the stock market and biology companies, there's this little company in California called Gilead that basically turned HIV into a chronic illness, is cured hepatitis C, and is close to curing hepatitis B. And that company had two concepts, okay? And first of all, this company is not run by lawyers and chartered accountants. It's run by chemists. And their thought is that with good chemistry, and we have the best stereochemistry in the world, we can produce drugs that have just as high affinity as monoclonal antibody that are small molecules. And second concept is you don't cure complex disease processes with one drug. So the polypill, dual, triple therapy, I think, is the future of IBD in small molecules. And that's going to take 10 years, but that's where we're going. So here are the classic sort of beliefs about monoclonals versus small molecules. So monoclonals are highly specific, and they have minimal risk of off-target side effects. That's their major advantage. The limitations, however, are that these are large proteins, they're immunogenic, and you have to give them parenterally. Small molecules, on the other hand, can be given orally and do not carry the risk of immunogenicity. However, off-target side effects are a rate-limiting safety concern, and the cost of small molecules is less. All sounds good. Well, this is challenged. So monoclonals are highly specific. Well, you can design, as I mentioned, good small molecules that have just a high avidity for target as monoclonals. The limitations of immunogenic. Well, you heard Scott this morning, don't worry about ustekinumab. The immunogenicity is very, very low, so they're not all immunogenic. And there's a large amount of work being done now about giving proteins orally and delivering them to the gut and getting large molecules across the mucosa. And then small molecules, what about the off-target side effects? Well, it's true, but the risk can be mitigated in many cases. Cost of small molecules is less? Well, not always. The price of TOFA is substantial. So it's a more complicated world than those statements that I had up initially. The good news is that, at least in ulcerative colitis, step care is alive and well, and there's many opportunities for small molecules at any step in the pathway. So I'm going to talk about 4 classes of drugs. I'll briefly go back to the JAK inhibitors, S1P1 agonists, phosphodiesterase 4 inhibitors, and oral peptides. So let's start with the JAKs. So the cytokine pathways in ulcerative colitis we've heard about before, and the pathogenesis. And the kinase inhibitors are really docking receptors. These are transmembrane G proteins. And this is this fine-looking fellow with 2 faces is Janus, the Roman god of portals and doorways. So January, looking in to the new year and looking back at the old year. So this is Janus, and so the analogy with this protein, they're transmembrane, so they sample the outside world and they signal to the inside world. There's a variety of these compounds. Everyone has a JAK and everyone has a favorite JAK, and Scott showed you the data, so I'm not going to spend much time with it. The drug certainly is effective. These are the maintenance data. And you heard that the FDA advisory panel made a recommendation to approve, or specifically favored that the drug's therapeutic index was good. I think the problem with JAKs is that these are broad-spectrum immunosuppressives. So this is like azathioprine that really works, okay? So, and you can expect to see all the things that go with that. So there's a very strong signal for varicella zoster. If you look at the risk of varicella zoster, age is the biggest risk factor. If you take a 60-year-old, their risk over a year of developing varicella zoster is about the order of 3%. If you're on a TNF blocker, you double that, 6%. If you're on TOFA, you double it again, it's 12%. So the new vaccine is a very important advance, and we've got to figure out how to pay for that and get payers to pay for it, but that will solve that problem. And Scott alluded to lipids and some other things. But these are very useful drugs. My own view is that they're not going to be first-line agents. These are going to be good salvage drugs. And I mentioned earlier this morning, we've got a big problem with if you fail a TNF blocker. So this is useful there. Here's a really interesting approach. So Theravance is a small biotech company, and they have an oral pan-JAK inhibitor. And so this is something that is poorly absorbed, and the notion there is you're going to get a topical effect of the JAK, and this may be a solution to the systemic immune suppression. So tune into this phase. This is running a phase 2 study right now, and you'll see results from it fairly soon for the future. Now, here's a class of agents that is going to come to the clinic for sure. So this is the concept of modulating S1P1 receptors, and you saw the fancy cartoon about trafficking inhibitors. They're very specific mechanisms of how T cells get into the gut. Well, think about it for a minute. If there are those very complex mechanisms to get T cells into the gut, there has to be a complex mechanism to get them out of the gut. And this is how it works. Essentially, the T cells have receptors on them that recognize sphingophosphates, essentially lipids with phosphorylated ends to them. And these lipids are expressed on the endothelial cells of the lymphs, and there's a gradient that goes from low concentration of the residues in the center of the lymph node to increasing concentrations. And the lymphocytes use this receptor to follow the phosphate residues and to exit the lymph node. What this class of drugs does is it internalizes the receptor so the lymphocytes can't feel the gradient, so they get stuck there. You remember when we used natalizumab, when we were doing the clinical trials, we had to be blinded because the lymphocyte counts would go up 40% on average because you're blocking everything. When you block alpha-4, you block alpha-4 beta-1 and alpha-4 beta-7 reactions. This is the opposite of that. The lymphocyte counts go down about 40% because they're being trapped in the lymph nodes. Fingolimod was the first member of this class. It's a breakthrough drug for multiple sclerosis. It's about half as effective as natalizumab, but it's more effective than beta interferon or Copaxone. So it's been a really big advance. Fingolimod's a pretty dirty drug. A lot of problems, cardiac toxicity, interstitial lung disease. This agent that I'm going to talk about, ozanimod, has been cleaned up, so it's more specific for S1P1. This is the first study, and you can see, arguably, while only a 10% difference at week 8, in the most effective 1 milligram dose. But look at week 32, this is a drug that I think is slower acting, but has legs. And the toxicity profile was very good in the phase 2. The phase 3 study is now 3/4 recruited, and we'll have a result from this in about a year and a half. And just actually last week, there was a second S1P1 antagonist that's been studied that released positive phase 2 data, and the effect size looked just like this. So almost certainly this is going to be a new drug for ulcerative colitis. We really don't have data yet in Crohn's disease. And this is just showing some open label extension data from that Touchstone study, and the message being essentially that when you cross the patients over who are on placebo or the ineffective dose, 0.5 milligrams, you can see that in their partial Mayo scores, they look like the patients that were treated with the 1 milligram over time. So this is a really, I think, positive study and optimistic. And as I mentioned, the side effects, the big one we worried about was bradycardia, because there's actually, it turns out that there's expression of the receptor in the conducting system in the heart. But this has been cleaned up by making it very specific for the type 1 isotype of the enzyme. So apart from some asymptomatic liver enzyme elevations, it was really a pretty clean safety profile. I'd like to now move on, Premolest and ulcerative colitis. This is a phosphodiesterase 4 inhibitor. Unfortunately, it doesn't cross-react with phosphodiesterase 5, at least for the male population. That would be a good side effect, but it's specific for phosphodiesterase 4, which kind of, how it actually works is it downregulates expression of cytokines in a global fashion, not to the level that you get systemic immune suppression. And this has been a big success in dermatology. Those of you who are television aficionados would recognize that young woman that's dancing around with her black dress and going to parties now that she's on Premolist. Her psoriasis is under control. If you look at PASI scores, at the bottom there, showing the data from the pivotal studies, drugs that are really effective in psoriasis, such as ustekinumab, or I mentioned the new 12 to the 23 inhibitors or anti-IL-17s, they get PASI 90 scores or complete clearing scores of north of 75, 80%. That's not this. These are actually PASI 70 scores or 75 scores, and you can see that you're getting right 45, 50%. So these drugs are not highly effective anti-psoriatic agents, but they're completely safe. And so this has turned into $1 billion drug in psoriasis therapy. And we now have, as of ECHO, this result. So this is a phase 2 study in ulcerative colitis, remission, Mayo score, endoscopy plus clinical symptoms. And you can see 31.6% for 30 milligrams BID versus 13.8 for placebo. Now, those of you who are astute about this study would say there's just one little problem with this study. Small molecules, unlike monoclonal antibodies, generally show proportionality from an exposure standpoint. And the high dose is actually lower, the point estimate, than the low dose. So that was disturbing and hard to explain, but to me, this is a clear-cut signal, and this molecule is now going to go into phase 3. And so where could this drug be positioned? Anywhere you like, okay? So, but milder disease. And, this is ulcerative colitis, so after 5-ASA, this could fit in here. In Crohn's disease, we don't have an effective, well-tolerated oral therapy to use as first line. And if this is borne out in Crohn's disease, it could enter there as well. And then just really futuristic, I've talked about oral peptides. Remember cyclosporin? Cyclosporin is a cyclic peptide. And because it's cyclic, it's resistant to proteases in the gut. Well, you could do the same trick, configure the amino acids differently, and you could end up with drugs such as this one, oral alpha-4 beta-7. This is a phase 2 study has finished recruitment, and we will see data. So an oral alpha-4 beta-7, well, you'd expect it to be safe. It's targeting alpha-4 beta-7 is safe. And if it can get around the need for parenteral administration, this could be a real game changer as well. And this doesn't have to be restricted to alpha-4 beta-7. You could design one of these peptides to actually target TNF or interleukin-23. And you can imagine that you might mix several of those oral peptides together and have your first generation combo therapy biologic target. Okay, so I'm going to conclude there. I've talked about small molecules. There's multiple new classes of agents under development. The JAK inhibitor is going to be here within a matter of months. And the S1P1s, I think, are going to make it. We've got a really strong signal with phosphodiesterase 4, as in psoriasis. And I've talked about the oral peptides, and the landscape's changing. And I won't say we're going to be as confused as rheumatologists, because they're really confused. They've got about 9 biologic drugs and half a dozen small molecules, and ask them to write out a treatment algorithm, and they just throw up their hands. We're going to do better than that, but time will tell. Thank you.